Well, good afternoon. I would like to welcome you all to this ceremony. And I would like to officially open this academic ceremony in which Ms. Claudia Schönichen will defend her academic thesis entitled A Jack of All Trades, Platelets at the Interface of Endothelial Inflammation and Coagulation. May I invite you, Ms. Candidate, to present a summary of your studies and the conclusion of your thesis in the next 15 minutes. And I'd like to give you the word.
Dear Pro-Rector, dear members of the Assessment Committee, dear colleagues, family, and friends. In the next 15 minutes, I would like to give you a brief overview of the aims and main results of my thesis, the research I conducted in the past 5 years, and I hopefully show you some of the many fascinating aspects of platelet biology. I want to start by introducing platelets, one of my favorite types of cells. Platelets are the smallest blood cells, and together with the coagulation product fibrin, they form blood clots. This is crucial to stop bleeding upon a vessel injury, a process called hemostasis. In this process, they change their appearance from disc-like, resting, to activated, where they form long, tentacle-like protrusions, eventually resulting in a three-dimensional thrombus. Beyond hemostasis, platelets play an important role in immunity and wound healing as well. If blood clotting happens without the injury of a blood vessel, this is called thrombosis. This happens, for instance, in a life-threatening conditions such as stroke in the brain or in a heart attack. In these conditions, blood clots block an essential blood vessel, stopping the supply to the subsequent organ. 1 in 4 deaths worldwide are because of a thrombotic event. Therefore, understanding platelet function is crucial for prevention and the treatment of these diseases. But how do platelets know when to activate? To monitor and communicate with their surroundings, platelets have sensors on their surface, so-called receptors. For instance, activating receptors and adhesive receptors. In addition, platelets contain large amounts of granules that they can release when necessary. When a blood vessel is injured, this provides a trigger for the activating receptors of the platelets, also called a ligand. When a ligand binds its receptor, the platelet rapidly changes its shape, contracting its core and forming long finger-like protrusions. This goes along with more adhesive receptors on the platelet surface and the release of platelet granules. All of this will trigger the recruitment of other platelets and immune cells and eventually lead to the formation of a three-dimensional thrombus stabilized with fibrin fibers, sealing the injury. In healthy conditions, our blood vessels prevent the formation of blood clots, and responsible for this is the innermost layer of our blood vessels, the endothelium. The cells of the endothelium, called endothelial cells, constantly secrete inhibitory mediators that keep the platelets in a resting state. Further, endothelial cells inhibit the formation of fibrin, thereby preventing blood clots to form. In endothelial inflammation, a common underlying condition in diseases like stroke or heart attack, the endothelial cells can no longer sufficiently inhibit platelet activation. The activation of platelets and release of granules leads to the recruitment of more platelets and immune cells that can further enhance the endothelial inflammation. In addition, endothelial cells in inflammation can no longer sufficiently block the formation of fibrin. All of these processes lead to an increased risk of thrombosis in inflammatory diseases. However, how these mechanisms are altered in inflammation is not very well understood. So we have an incomplete understanding of platelet activation at the vessel wall, and therefore the aims of my thesis were the following. How does vascular injury determine thrombus formation by platelets and by fibrin? How does thrombus formation determine platelet activation and interactions with immune cells? Is there mechanisms or are there mechanisms that prevent unwanted platelet activation? And how does vascular inflammation influence platelet activation in these processes? The central method I used in my research is a microfluidic thrombosis model. In this assay, makes use of a small microfluidic channel with a glass bottom that contains an adhesive surface. When blood is perfused through this channel, platelets in the blood sample will encounter the surface and will sequentially activate until they form a clot, stabilized with fibrin fibers. This assay allows us to microscopically monitor thrombus growth, cell adhesion, and fibrin formation. It allows us to look at activation markers such as intracellular calcium and the integration of endothelial cells to measure thrombus formation and fibrin in a so-called vessel-on-a-chip model. I already mentioned that activated platelets communicate with immune cells through their receptors and their release granules. What this means in early stages of thrombus formation or in immunity is not fully understood. In chapter 2 of my thesis, I investigated how platelets in a thrombus in our microfluidic model can interact with immune cells in early stages of thrombus formation. Therefore, we use two tools. The first tool is blood from patients that have a condition called Glanzmann thrombocythemia. These patients lack the most abundant adhesive receptors on the platelet surface. These platelets cannot form 3D thrombi, but can otherwise be normally activated. The second tool is a pharmacological inhibition of platelet granule release. This is an example of a thrombus formed in this model. The small circular shapes are the platelets, and the fiber-like structure is fibrin. The big aggregates are the thrombi. When platelets are missing their most abundant adhesive receptor, no three-dimensional thrombi can form. In addition, we observed a large number of immune cells that adhere to a monolayer of platelets in otherwise the same conditions. We therefore conclude that platelet surface receptors limit immune cell behavior at a thrombus in healthy individuals. Further, we tested if platelet secretion would affect the behavior of the immune cells at the thrombus. For this, we tweaked our model to enrich immune cells at the thrombus by inducing a short period of stasis. We loaded the immune cells with a fluorescent dye that lights up when the cells get activated, shown in the rainbow colors. We can also monitor the movement of the cells, and in a normal secreting thrombus, we observe high activation levels in the immune cells and lots of movement. Interestingly, when we blocked secretion of the platelets in the thrombus, the activation signals were reduced, and we observed less movement of the cells. Therefore, we conclude that platelet secretion is important for the activation of immune cells at a thrombus. Together, these experiments show that platelets in a thrombus steer immune cell adhesion and activation through their receptors and granule release. In the next chapter, we wanted to clarify molecular mechanisms that regulate platelet activation. We already know that platelet activation goes along with shape change, contraction, membrane protrusions, and the release of granules. On a molecular level, when a ligand activates a platelet, this will lead to the activation of a protein called protein kinase C, a key player required for all of these platelet activation steps to properly form a thrombus. This also goes along with increased intracellular calcium level. So far, it is not known what the relationship is between protein kinase C activation and calcium increase in the platelets. Therefore, we measured calcium in activated platelets and used inhibitors to prevent protein kinase C activation. When we add an agonist to platelets, calcium levels in the platelets increase as shown in this graph. When we added a protein kinase C inhibitor, surprisingly, these signals increased by almost fourfold. Until now, protein kinase C is considered to be required for all platelet activation processes, but our results show that protein kinase C also suppresses calcium entry, thereby preventing unwanted activation of platelets. This may be an important mechanism to prevent the unwanted activation of platelets and thrombosis. In the last 3 chapters of my thesis, I investigated the effect of endothelial cell on platelet activation in the vessel-on-a-chip model. This time, the images show fluorescent colors of platelets in the thrombus and fibrin. The rainbow colors show the intracellular calcium rises in the platelets corresponding to activation levels, and in red you can see fibrin buildup. After 3.5 minutes, large thrombi form and platelets adhere to the surface, and we see that when endothelial cells are present at the same surface, the large cyan-colored shapes, thrombus formation is strongly inhibited. We only see residual calcium signals in the platelets, and in addition, we observe almost no fibrin formation. Endothelial cells therefore prevent platelet activation and fibrin formation under flow. We also tested inflammatory conditions by treating the cells with either TNF or LPS, 2 different inflammatory stimuli. We see that in inflammatory conditions, the endothelial cells are no longer to inhibit platelet intracellular calcium, thrombus formation, and fibrin buildup. Therefore, we conclude that endothelial cells are indeed less able to prevent thrombus formation in inflammatory conditions. We wanted to know what exactly is different in the inflamed endothelial cells, so we looked at the endothelial cell content using a method called transcriptome analysis. In this heat map, each small horizontal bar shows the levels of a certain gene in each of the 3 conditions. You can immediately see that 3 characteristic patterns arise, each corresponding to our 3 conditions: untreated cells and the 2 inflammatory conditions. When we zoomed in to identify clusters of alteration corresponding to pathways, but also individual genes that are changed in inflammation, we identified numerous pathways that are altered, which we could link to platelet activation and fibrin formation. This means that in inflammation, endothelial cells are less able to prevent blood clot formation, contributing to an increased thrombosis risk in inflammatory diseases. With this, I would like to summarize my findings. I hope I could show you that activated platelets, on one hand, restrict immune cell adhesion through the adhesive receptors, and on the other hand, support immune cell activation through the release of their granules in our microfluidic model. Activated platelets require protein kinase C to secrete and aggregate, but at the same time, they use protein kinase C to limit calcium entry, preventing unwanted thrombosis. Lastly, I showed that endothelial cells prevent platelet activation and fibrin formation under flow, and that this prevention is partly lost when the endothelial cells are inflamed via several different mechanisms. I would like to leave you with 2 messages for today. The first one is that platelets are very diverse cells. They are involved in multiple intracellular interactions, making them a true jack of all trades. And secondly, understanding platelet activation mechanisms at the vessel wall is crucial for the management of thrombotic diseases, especially in inflammation. With this, I would like to thank you for your attention, and I hand the word back to the prorector.
Thank you very much, Ms. Candidate, for your clear presentation and well illustrated, and I think it offers the opportunity for quite some questions. But before we start the questioning, I'm going to introduce the promotion team who sits opposite of me, and your promotion team has guided you the last 4 years and has brought you to what you're standing today to defend your thesis. And opposite of me, and I start from my right side, so closest to the audience, is Dr. Jurk. Dr. Jurk is Associate Professor of Platelet Physiology and Function of the Center for Thrombosis and Homeostasis at the Johannes Gutenberg Universiteit in Mainz. Welcome, and also thank you that you came all the way from Mainz today to this event. In the middle is Dr. Kuipers. She's assistant professor at the Department of Biochemistry at our university. And the first promoter is Professor Heemskerk, and he's professor of cell biochemistry, of thrombosis and homeostasis. And this team has guided you in the last 4 years. For the audience, we have 6 members in the Corona, 6 members who are going to address questions. 4 of them are here on site, we call it, present here today, and 2 will be online. They're on the screen, but later you will see them back here on the full screen. Saying this, we're going to start, and as usual, we're going to start with the chair of the assessment committee, and I would like to welcome Professor ten Kate, who is professor of clinical thrombosis and homeostasis at our university, to give him the opportunity to start the discussion.
Thank you, Mr. Prorector. Dear candidate, first of all, I'd like to congratulate you with the accomplishment of this very nice thesis. I like the title very much, Jack of All Trades, and also the cover. It's beautiful, beautiful work, and also the contents is quite nice. I want to share the supervisory team in the congratulations. So now it's time to defend it. For a start, I wanted to kick off with discussing one of the propositions, number 5. So maybe one of your para-nymphen will be kind enough to read it.
The advent of mass spectrometry-based omic technologies, such as proteomics, metabolomics...
Oh, I'm sorry, number 5.
Sorry. Inflammation and thrombosis are closely linked processes, with platelets playing a crucial role in both. This recognition underscores the need to identify new platelet-directed therapeutic targets.
Excellent. Well, as the prorector reminded us, propositions should be defended. So I intended, in first instance, to ask you a question. So which platelet-derived or directed therapeutic targets are you considering? But that's too simple. So maybe we should come to a bit more interaction. So maybe I could ask a broader question, why platelets? Well, you point them out, it's a jack of all trades, and they're extremely important, but if you try to knock out platelet targets, there is always a concern that you introduce a bleeding risk in a patient because the platelets are so important for hemostasis. So which targets would be safe targets, in your opinion?
Highly esteemed opponent, thank you for your compliments and your question. Yes, of course, with platelets playing such an important role in hemostasis, you have to make sure if you target them that you monitor or minimize the bleeding risk. But platelets have a lot of immune functions, and we know that processes like thromboinflammation and immunothrombosis are partly mediated through platelet activation, and therefore it's best to tackle the immunological side of the platelets. So one thing that one could think about is chemokines or chemokine receptors on the platelets that are known to not fully activate the platelets but play more of a supportive role in the platelet activation. So that would be one way to go. Other options are GP6. We know that GP6 knock out patients. You don't say that, but people with a genetic dysfunction of GP6, they tend to not have bleeding problems. So that could also be another avenue to go.
Yeah, and GP6 is already, under development. The clinical studies are being done with commercial reagents, and that looks promising indeed. And which chemokine receptors would you consider? Because chemokines are not very platelet specific, right? I mean,
that's true. If you dive into the literature, there's a lot of conflicting evidence which chemokine receptors are indeed on platelets, and whether or not this expression level corresponds to a functionality. We never did those experiments, but in actually the discussion, there are, I highlighted a bunch of the receptors that could be targeted on page.
You did. That was in the general discussion.
I think so, yes. So we know that platelets, for instance, can react to CCL2, and they also release other chemokines. So, receptors for that.
And would that be a sufficiently safe approach in terms of immunology?
That's a good question, because a lot of the chemokines also have some sort of a dual role in not only thrombosis, where it's maybe not so clear, but they often also play an important role in vascular remodeling after ischemia reperfusion, for instance. So that would have to be evaluated.
Yeah.
So it's a bit tricky with the chemokines as well, because immunity is very complex.
It is.
Yes.
Yeah. And that's what we have seen in studies in anti-IL-6 and so. Is CLEC2 a target that you would consider?
Potentially.
Yeah.
Yeah. I mean, we know that it's very important in lymphatic development. I'm not fully sure what knockout studies or conditional knockout studies show or indicate, but it could be a target.
Yeah.
Yeah, for sure.
Okay. Thank you very much. Maybe I can move then to chapter 6.
Okay.
Chapter 5 and 6, or chapters 4 through 6, are quite interesting in looking at UVEX, and that's a very old strategy, I think, because in the past many studies were done, experimental studies in vitro, in looking at pro-inflammatory mediators and the impact on endothelial cell anticoagulant properties. And from those days I recalled, and therefore it triggered my attention on page 250, where you discuss endothelial heparin sulfate. And at the time when I was a postdoctoral fellow, long time ago, I was in Boston working with Professor Robert Rosenberg, and he was one of the investigators that knocked out heparin sulfate, at least the enzyme that's responsible for generating active heparin sulfate on endothelial cells. And it was a paper in the Journal of Clinical Investigation. And it showed surprisingly that it did not have any impact on hemostasis, on the anticoagulant function in mice, under in vivo conditions. So whereas in UVEC you could probably neutralize the anticoagulant effect, but the mice were quite viable and didn't have any increased fibrin formation under conditions of hypoxia and so. So I was wondering whether the polybrain in your system, you still think that the heparin sulfate is the most important target, or would there be other glycosaminoglycans that are more important in interacting with antithrombin? I realize it's a very specific question. It's but
yeah, it's a very charged molecule, and molecules of the glycocalyx seem to they are also very charged. So it would be, or I would think that polybrain can bind to all of the proteoglycans in the glycocalyx. So I don't think it's very specific.
No. Yeah,
so that's definitely a possibility that other proteoglycans in the glycocalyx are an important player as well.
Yeah. I agree. I'm very happy with your answers, so I'd like to give the word back to the prorector.
Thank you. And then we move swiftly online to Professor Reinhardt. Professor Reinhardt is Professor of Knotobiology, Thrombosis and Hemostasis at the University Medical Center of the Johannes Gutenberg Universiteit in Mainz, and I would like to welcome him. And I'd like to give him the word to ask questions online with you. Professor Reinhardt.
So thank you, Prodekan, for handing over the word to me. And I am impressed by the really nice thesis of Claudia Schönichen. And I was really interested in chapter 2 and on the role of alpha-2b beta-3 in neutrophil adhesion and also in the migration behavior of leukocytes. So how is that actually mediated? I mean, you looked on chemokine effects, but what would be the molecular basis of those leukocytes and neutrophils in the migration? Can you explain a bit which molecules might be relevant and how this could be mediated?
Thank you for your question and your compliment. Oh, there's an echo. Can you hear me?
Yes.
Okay. So in our observation, we saw that integrin alpha-2b beta-3 somewhat prevents the adhesion of neutrophils to a thrombus. And we could show that also pharmacologically by post-perfusing the thrombi with tirofiban, for instance. And then we would see an increase of neutrophils to the thrombus. I believe that the strongest stimulant of neutrophils at a thrombus are most likely also the chemokines. And we know that platelets can release a lot of chemokines, and we actually tested them, and we found that CXCL7 was the strongest inducer of calcium in the neutrophils and would also induce very strong movement patterns in the cells in an isolated system. So my guess is that a combination of chemokines, and in our hands mostly CXCL7, would be the strongest activator of neutrophil activity at a thrombus, whereas the adhesive receptors somewhat prevented the neutrophil adhesion, at least in the early stages of thrombus formation and in flow conditions.
Right. So is there something like a matrix molecule needed for this migration behavior on the platelet thrombus? Do you have an idea on that, or could that be tested and addressed?
Actually, one of the suggestions we got from the reviewers is test this in fibrinogen deficient plasma. But we were never able to really do those reconstitution experiments because they are technically quite challenging. Fibrinogen binding could be one thing that prevents maybe leukocyte attachment to the thrombus. But we also saw that neutrophils would kind of favor phosphatidylserine exposing sites of the thrombus. And we do know that there's an interaction here. So because it's such a delicate balance also with neutrophil activation, it is possible that platelets kind of mediate their behavior rather than fully activate them at any given thrombus, because that may not be favorable.
Right. So thank you for addressing this aspect. I have maybe another aspect on chapter 4 of your thesis work, and there you looked on PAR activation. And of course, there's also PAR activation and PARs on the vascular endothelium. Does the endothelium also contribute to this activation process? And would you be able to dissect this PAR activation on the endothelial cells from that based on the platelet activation process? Is there a way to dissect that, or what would you propose here?
That's an interesting aspect, and of course something I would also be interested in looking at. In using the omics studies, it's a bit difficult to really dissect it after the microfluidic model, because if we perfuse platelets over the endothelial cells, it is very difficult to dissect afterwards which proteome signature would belong to which cell type. What we can do is specific cellular probes, and one way to tackle that in proteomics could also be the SILEC method, where you can label the endothelial cells with a heavy isotope, and then afterwards you could perfuse the platelets through the channel, make a thrombus, and then really see what are the changes in the platelets, what are the changes in the endothelial cells. However, with plasma present, that's a bit more difficult, because plasma will contribute to a lot of background in your proteomics studies. So proteomics is easier the more pure your cell population is, and therefore is a bit more tricky, because perfusing just platelets over the cells won't give you a lot of thrombus formation in a way, because you need the other blood constituents too.
So thank you. So this was addressing my question, and I think it's a really very nice aspect of your thesis work to separate that out and to describe that.
Then the opposition will be continued on site. We move back here to the aula and we move to Professor Uri. Professor Uri is Professor of Cardiovascular Sciences of the Giga Institute at the Université de Liège in Belgium. And I also would like to welcome her and thank you that she joins us today. And I'd like to give her the word.
Welcome. Thank you, Mr. Prorector. So I would like to recongratulate Mrs. Candidate for the quantity and the quality of the work. That's impressive, the number of publications that you could have during your 4-year thesis. So congratulations. I will have, the first question will be rather mechanistic questions. It's related to chapter 3 and by the calcium increase that we can observe in platelets upon activation. So you nicely explained that in terms of calcium increase, you have a first step, which is the release of the intracellular pool from the stores, and then you activate the store-operated calcium entry. And you also say that we also have different kinds of channels, like the P2X1 channel and also sodium calcium exchangers that are also involved and not part in these 2 steps of calcium increase. So when you present the fact that when you inhibit protein kinase C, you further increase the calcium increase, what is the link between that and the calcium entry that does not depend on the release of the stores and on the SOC? So in other words, what is the link of this between protein kinase C inhibition and calcium entry through P2X1 or through the sodium calcium exchangers?
Highly esteemed opponent, thank you for your compliments and your question. That is a very good question, and actually, quite honestly, not something we investigated. And maybe the reason for that is because in our proteomic data, we did actually see, we could identify phosphorylation sites of the proteins that are involved in the SOS, in the store-operated calcium entry. So we did a phosphorylation prediction of BIN1 and STIM2, or the other way around, BIN2, STIM1. And we could actually identify a very likely phosphorylation site of protein kinase C on both of these proteins. Therefore, this is the pathway that we suggest and also investigated. In addition, our studies with the not the knockout patients, but the patients with the genetic mutations show that this pathway is indeed released to the store-operated calcium entry. How protein kinase C relates to the other calcium channels, quite honestly, we didn't investigate in this study, at least. But yeah, there's definitely room for that, of course.
I know it was a difficult question, but okay, we are there to challenge you. So, and also, you nicely say that when we have a prolonged calcium increase in platelets, it tends to leads to exposure of phosphatidylserine, so the creation of procoagulant platelets. And is this link clearly established? That is the first question. And what kind of other calcium fluxes would lead to a pro-aggregatory function for the platelets compared to the procoagulant platelet activity?
Sorry, can you repeat the last part of the question?
So when we have prolonged calcium increase, it's established that, is it really established that this leads to phosphatidylserine exposure on the platelets, so production of procoagulant platelets? Yeah. What do we know about pro-aggregatory platelets? Oh. The link between platelet aggregation and the kind of calcium flux that we can have in the platelet. Yeah.
So in our hands, at least, it's quite established that prolonged calcium increase in the platelets will lead to the formation of procoagulant platelets. And this is also a mechanism that we seem to observe in the thrombin generation assay that we performed later on, where we had the thrombin generation, and when we add CRP to platelet-rich plasma and really fully activate them, then we see more thrombin generation, and this is most likely linked to the phosphatidylserine exposure and prolonged calcium. In the experiments that we performed, we really think that the activation of protein kinase C leads to a more aggregatory phenotype of the platelets, kind of preventing this phosphatidylserine exposure and a little bit preventing this procoagulant phenotype. This is the mechanism that we suggest, based on the various different experiments we performed.
Okay, thank you. So another couple of questions will be about the chapter 4 and all the mechanism of endothelial-dependent platelet inhibition. Because you know that in the field of biomaterials, people try to reproduce this inhibitory activity of endothelium by creating some kind of coating on top of the biomaterials to prevent thrombotic complications. And you explain us that, and you show in your thesis that probably that the mechanism depending on nitric oxide are not the only one, sure, that inhibits platelet. So don't you think? And knowing that a lot of coating that are being developed now are based on nitric oxide release. So do you really believe that this will be sufficient to prevent thrombotic complications related to biomaterials in coatings?
My guess would be it's potentially not sufficient to really block the platelet, to fully block the platelet activation, because in our model we do see that there's several mechanisms, and especially in our model, nitric oxide didn't seem to play that much of a role. And when we look at knockout models in mice, for instance, of enos, then it also doesn't seem to be the major complication that they have thrombosis. The major complication these animals get is a vascular complication, so they get alteration in the vasculature rather than in the platelets. So my guess is that if you want to coat biomaterials, that you may need a combination of inhibitory mediators, and one idea would be maybe try to mimic the glycocalyx and all of its components and not just one of them. In addition, nitric oxide is maybe also a bit too short-lived to really work that in a big system like ECMO, for example.
Very interesting, thank you. And the very last one, or is it over?
If it's a short one, yes.
Okay, then I will leave it like this. Thank you very much.
Okay, thank you.
Thank you very much. Then the opposition will be continued by Dr. Nuls. She is Associate Professor in the Department of Biochemistry at our university, and I'd like to give her the opportunity to discuss with you.
Thank you, Mr. Prorector. Dear candidate, congratulations with this very nice work. It's not just a lot of work, but it's also very interesting to read and very interesting findings. And I want to discuss with you a little bit more in detail your chapter 2, where you investigated the interaction between neutrophils and thrombus formation, where you use this very nice setup, this microfluidic unit, where you investigate thrombus formation on these collagen-coated slides. And you see indeed neutrophils adhering, but not so much the nets you mentioned at some point. So first of all, why is it interesting to look at that? Is it useful to have a look at that? Or, and secondly, you didn't see that. Is that now a problem, or what would you do as next steps?
Esteemed opponent, thank you for your question and your compliments. Did I understand the question correctly, that it's about the nets and why we don't see them? Okay. Yeah. I believe that for net formation, because it's such a potent biological process, let's say, because it's very pro-thrombotic, procoagulant. So I would think that you have to be careful when and how to produce nets. I think one aspect that's missing in our model is the vascular damage. So we do not have damaged endothelial cells that we would have, for example, in the cremaster injury model, where we see endothelial damage, and then also neutrophils really rapidly adhere to the injury. So this is missing in our platelet model because we only have the microfluidic channel. In addition, I believe that in the arterial flow conditions, maybe a lot of the activation that would in venous conditions be there are flown away. They're actually maybe just float away, and that's why when we induce a stasis, we see the adherence of neutrophils, but maybe it's still not enough to really induce this net formation. Another possibility could be that platelets actually inhibit net formation, and there are, I think I found one study that investigated a very specific population of CD40 ligand positive platelets that could inhibit or would inhibit neutrophil activation and net formation. So that's possible.
Yeah, okay, thank you. So you also mentioned the vascular damage, but so you also did experiments with endothelial cells, UVEC cells. So there you did some stimulation experiments on inflammatory conditions. Do you think that you examined there also effects on whether more neutrophils were adhering or net formation, or what would you expect there?
To be honest, we bluntly ignored that in the platelet activation experiments. That's because of the scope of the paper, but I would expect that we do see, of course, neutrophils adhering on inflamed endothelium just in the bright field. We see that. We didn't specifically look at that. One problem with the net assay is that you have to incubate the chambers relatively long, and we had a lot of issues post-staining the nets in the perfusion chamber because they are not as stable in our chamber. So it's for sure possible and probably also really interesting to look at, but we never had the chance to.
Okay. So yeah, as you said, so these nets, maybe they need a much higher trigger before you really get these, but you do see neutrophil adhesion. You did some in chapter 4 or 5. You also had a look to endothelial cells and when they are inflamed, how their secretome is changing. Was there maybe something in this secretome that maybe could lead to additional activation either of platelets, neutrophils, or their interaction? Might this also have then this influence, what you said, the effect of endothelial damage?
Yes. So I believe that we see, and unfortunately it's not in this version of the chapter, but in the submitted version, there's a list of secreted proteins from the endothelial cells that are potentially very interesting. And among those are more chemokines, but you also see increased levels of adhesion molecules that can, of course, also promote. But maybe it's just the combination of really a cocktail of chemokines that really signals to the neutrophils, okay, here we need some help. We need activation.
Okay. And maybe also in that direction, could it also be that you might need, I don't know, bigger thrombi, more activated thrombi, that might also increase neutrophil binding or activation? And is there a way to do that in your setup? I wouldn't know how, but you're the expert in this. So suppose that I would want to study this. Is there a way that I can somehow get bigger thrombi and then study interaction?
So bigger thrombi in this channel is a bit challenging because they are fairly small and you get fully blockage of the channel at some point. Mouse thrombi aren't necessarily much bigger than the thrombi we make in the channel. So maybe it would be an idea to look at mouse blood in this microfluidic channel and see if we would see neutrophil net formation there. We personally never did that. But that's an option. I don't think it's related to the size of the thrombus, honestly, but it could be really the amount of chemokines that are released and also attached maybe to the thrombus surface.
Yeah, okay, thank you. Then maybe one shorter question is, you see neutrophil binding, but you also mentioned that you have to prevent, or that you have to do a flow interruption at some point to see this, because then you can reduce the impact of this platelet integrin that you find. Why was that? So what do you think is the reason that you need this flow interruption to do
I really believe it's the amount of chemokines that can be present at the thrombus, because through this flow disturbance or slowing down of the flow, we of course decrease the shear rate, and chemokines also have the tendency to stick to molecules on thrombi or matrix proteins in general. So potentially the chemokines had the chance to stick around and therefore be more attractant to the neutrophils.
Yeah. Could it be also like flow interruption, that it could be a trigger for cell stress, maybe on platelets or neutrophils, and then maybe give some additional activation? Could that be also an option?
Could be an option, yeah. We didn't really look at that so much, but it's an option. One other option is also we never actually stained after the stasis. Could be more phosphatidylserine, but we didn't check for that.
Okay, thank you.
Yeah.
Very satisfied with the answers, and I give the word back to Mr. Prorector.
Thank you very much. Then the opposition will be continued now online again, and we go to Professor Schäfer. She was Professor of Translational Vascular Biology at the University Medical Center of the Johannes Gutenberg University in Mainz, and I also like to thank her that she joins us today online, and I'd like to give her the opportunity to discuss with you. Professor Schäfer.
Thank you, Mr. Prorector. Dear Ms. Candidate, also from my side, congratulations to this very detailed work, and I think with your publications and also the thesis, you really advanced the field, or a great amount of knowledge about the platelets and thrombus formation has been gained. Nevertheless, I have a few questions, and I'd like to most of them are on the endothelial part of your work, and maybe I start with one which relates to chapter 4 and 6, and where you describe the surprising antithrombotic phenotype of endothelial cells in your model. And you use human umbilical vein endothelial cells, which is a very much used type of endothelial cells in vascular biology. But of course, they're from the umbilical vein endothelium. And we also know there are different endothelial cell sources, and they differ also between organs. So can your findings, if you translate them now to the in vivo situation, and can you a little bit discuss this with us? What you think, how would this translate to different endothelial cell types, to different forms of thrombi, different diseases?
Highly esteemed opponent, thank you for your questions and your compliment. Yes, so of course HUVEC are HUVEC, and everybody who works with endothelial cells asks that question, why did you pick HUVEC? And the reason we picked HUVEC is indeed because they are very well characterized, and I'm aware that umbilical cord thrombosis is very rare, it practically never happens. But what we do seem to appreciate from this model that it is a bit of a hemostasis model rather than a thrombosis model. And we do see that if we treat it with the inflammatory stimuli, we can induce this pro-thrombotic and procoagulant phenotype in this model. If I were to do it again, I would probably also go for other cell types. And one that's quite important, to me at least, is the microvascular cells that I would maybe expect to behave differently. Because we never did those experiments, I'm not 100% sure what to expect or what I would expect from these cells. But one thing that I do know is that because we have the transcriptome analysis, we know very well what we're dealing with, and that is a big advantage of using these cells. So if I were to do it with other cell types, I would also make sure to really characterize these cells, maybe transcriptome, maybe proteome, to really understand what are the molecular mechanisms behind whatever cell type you're looking at.
Yeah, because they also differ in the expression of anticoagulant and procoagulant factors, so it could well be. Mm-hmm. Staying with the endothelium and your observation, I think in chapter 5, it's that you say endothelial cell factors, they induce like about 255 changes in the phosphoproteome in platelets. And did you have a chance not only on the phosphorylation, but also dephosphorylation, and which type of phosphor residues they are, and if there may be a target or platelet phosphatases, and which ones?
Yeah, so we didn't look at phosphatases that much. I kind of expected that questions, and I know your work on PTB1.
It actually is the most highly expressed platelet phosphatase.
So I'm aware that we kind of bypass the phosphatases in the work a little bit. What we can see, of course, is whether or not a phosphocyte is more abundant after exposure to endothelial cells than compared to when they're not. If that is because of dephosphorylation or because it's never been phosphorylated in the first place, it's a bit difficult to say. So we didn't investigate that in particular.
And what would you speculate if you expose like platelets, healthy platelets to inflamed endothelial cells, or inflamed platelets to healthy endothelial cells, and vice versa? How would you? What would you predict your changes?
So in the end, this is what we did in the second phosphoproteome analysis, where we treat our HUVEC with the inflammatory stimuli, and then we add platelets from healthy donors for half an hour, then stimulate them, and then we perform the phosphoproteome analysis. So this is exactly what we did with healthy platelets exposed to damaged endothelial cells. We didn't do it the other way around, and this is something that is definitely something I would do in the future, also in the context of thromboinflammation, to really see what do platelets do to endothelial cells and how does that contribute to the inflammation, because this is an aspect we didn't really investigate it.
Mm-hmm. And you focus on inflammatory stimuli like TNF and LPS, which would occur in vivo, like more in septic conditions. Maybe did you look at other stimuli, not that are more like cardiovascular risk factors? Or if I know you didn't, but which ones could you imagine?
We didn't, we limited to 2 different inflammatory stimuli, and in the end it worked out nicely because we have one very strong one and one that is a bit more mild, and that's also a nice comparison to have. If I were to do it again, I would probably also look at interleukins, interleukin-1 beta, for example, because that's also an interesting target in cardiovascular diseases. So I would probably opt for that if I had to set this up again.
Mm. Yeah. Okay, and then your ATVB paper and chapter 2, you show the data that the different strengths of the effect of the 3 different chemokines, CXCL7 and CXL5 and CXL4, and grade them and say they are like the magnitude of effect. I would challenge you and say this is an in vitro observation, and in vivo, would you think it would happen this similar way? And if yes, why? What would be a mechanism about this, or the sense also?
Mm-hmm. Yeah, that's a very good question, and I would have to speculate on that. And indeed, the models that we use is specifically with the chemokine activation of the neutrophils. That's a very isolated system, and I wouldn't bet that you would see the same findings in the in vivo situation. So that is something we would have to evaluate, maybe in a mouse model, or with certain inhibitors, but I don't know.
But for example, could it be that if it were like this in vivo, would it mean like you need a certain amount of sources, cellular sources, or would it depend on, I don't know, the receptors, or?
I have to close this discussion.
Okay. Mm-hmm.
I'm sorry, because I would like to give the opportunity also to the last opponent, Dr. Roest. He's affiliated with the Department of Thrombosis and Hemostasis of the Synapse Research Institute here in Maastricht, and I'd like to give him the opportunity. Dr. Roest.
Thank you very much, Mr. Pro-Rector. I just found out that I had the only chair without a microphone, so I took the one of my neighbor. Now, first of all, I like to give my compliments to your very nice thesis. It's really impressive, and I also like to share my appreciation to the promotion team. Well, I also like to go to chapter 2, and I like to start with the title, antagonistic roles of human platelet integrin alpha-2b beta-3 and chemokines in regulation of neutrophil activation and fate on arterial thrombi under flow. And my main question is, what do you mean by antagonistic?
Esteemed opponent, thank you for your compliments and your question. The reason we think it's antagonistic is because the activation of the integrin seems to really limit neutrophil recruitment to the thrombus. So platelets really prevent neutrophils from being at the thrombus at this stage in the model, whereas the chemokines will really lead to activation of the neutrophils at the thrombus. So therefore, we think it's a bit of a balance between recruitment of neutrophils and their activation and their steering their behavior at the thrombus.
So you see a direct role of alpha-2b beta-3 in the blocking of the binding of the neutrophils?
Direct, yeah, I think I mentioned it before that we never really looked at the binding partner on the neutrophils of this integrin. So we never tried to inhibit that in any other way. So that is missing, of course. But we do see the very strong effect with the Glanzmann patients, and we also see the same effect if we pharmacologically inhibit this integrin activation.
Yeah. So yeah, I completely agree with that. But still I want to know what is the explanation for this. So shall we go to figure 1 then? And if we go to figure 1, so my first surprise is that there's no surface coverage in the Glanzmann patients. Is that just because you have a measurement problem, or is there no platelet on the surface?
Are you referring to figure C?
Yeah, 1C, I'm sorry.
Yeah, so what we assessed there is this thrombus surface area coverage. So we didn't just look at platelet adhesion, because the platelets will form a monolayer on the collagen. That is something we can see, and that's also shown in the fluorescent images in the panel above. But if we look at thrombus, which is really the three-dimensional structure of a thrombus that I showed before, then we don't see that in the Glanzmann patients, and that is because they lack this fibrinogen alpha-2b beta-3 bridging between platelets. So the three-dimension thrombus is lost.
Yeah. Well, I'm very happy with that answer, because if that was not the case, then figure D was, then we could conclude that the leukocytes bind to collagen. Yeah. But my conclusion here is that the leukocytes are very good in binding to single cells, but not to aggregate. Do you agree with that?
Yeah, one idea could be that the less protrusions platelets have, maybe neutrophils have more access to binding sites. That maybe a steric problem, let's say. It could also be that, we do know that platelets have a lot of adhesive receptors, so, and we tested that later on with blocking the CD40 ligand and the p-selectin, for instance. So this could also be an alternative, that neutrophils just have the chance to bind to these other adhesive receptors rather than the integrin, because the integrin is so abundant in healthy platelets.
Well, that's a very good bridge. I wanted to continue on figure 1, but now you mentioned CD40 ligand and p-selectin or CD62P, then perhaps we can discuss that. Because, in the past I did a lot of platelet neutrophil interaction, and I did it under flow. And what I saw, if you measured real time, that on the aggregates, you very nicely saw the platelets rolling over the aggregates, but they disappeared again. So could it be that that's also the case in your study, that you just cannot measure the platelets that transiently adhere?
The platelets or neutrophils?
The neutrophils to the platelet aggregates.
Yeah, we didn't measure that, no. But we see it. We also see it in the flow model, of course. We didn't quantify it in the study, but we do see neutrophils, specifically in areas of flow disturbance, actually. So there seems to be something that triggers neutrophil adhesion. So if you're familiar with the microspotting, at the edge of the spot, for instance, where you have a bit of difference between the thrombus surface and the glass surface next to it, where you also have flow perturbations, and then you would see more neutrophils adhering to that.
Yeah. But what's then the explanation that in Glanzmann patients the neutrophils can stick, while in platelet aggregates the platelets do not stick?
Yeah.
The firm adhesion. Is that CD40 ligand? Or?
That's yeah, it could be. We found that if we block CD40 ligand, but that's of course including a period of stasis. So it's a bit tricky to answer that question. But if we do induce this period of stasis and then block the CD40 ligand, we see that neutrophils will stay less at a given thrombus, and they also will return back to the same thrombus less, which is here called fidelity. So we do see increase in movement if we block that interaction.
Okay, but if it's not CD40 ligand, do you think there's another receptor that can induce the firm adhesion?
PS.
That's a P.
You can a little bit elaborate more on the answer, but yes is sufficient, and you can stop whatever you want. It's your decision.
So I think PS exposure may be one of the options, but yeah, we never blocked that, so we don't know.
Okay, thank you.
Thank you very much. Dear Ms. Candidate, dear Ms. Schönichen, the time appointed for defending your thesis has now passed. The degree committee will now withdraw, and we're going to discuss not so much the quality of your thesis, that has been approved, but you're going to discuss the way you have defended your thesis this afternoon. I request that you and your company await the results of our deliberation and the return in this aula. Thank you very much.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. South side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
Dear Ms. Schönichen, the degree committee here present has assessed the quality of your thesis and in particular the way you defended your thesis about in the last 45 minutes. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you officially the degree of doctor. Professor Heemskerk is authorized to confer upon you this academic distinction in accordance with Dutch university customs, and therefore I would like to invite your supervisor now to take the floor, Professor Heemskerk.
So we first got a promise. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
Okay, that's good. By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Claudia Schönichen, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affix it with the official seal of the university. Dear Dr. Schönichen, my sincere congratulations also to your family with this first milestone, because it's another degree to go for. It was a wonderful journey. I liked it a lot. Lots of straightforward pathways, side pathways. And now I hand over to your co-promoter, Dr. Kuipers, for the laureate.
Esteemed Dr. Schönichen, dear Claudia, I'm very happy and proud that I am one of the first to congratulate you on obtaining your PhD. And also I want to congratulate your husband, Timo, your family and your friends with this great accomplishment. So this morning I learned that today is officially called Blue Monday. But to me it is not blue at all. I think it actually should be called golden, because it marks the end of your PhD journey, at least here at the University of Maastricht. You started your project in November 2009 as ESR 9 in the T-cardio... ESR 9 you are, right?
Okay, sorry,
of the
yeah,
T-cardio consortium.
No,
Yeah, right? Okay. Okay. But during your bachelor and master studies, you already gained a lot of research experience in the lab of Daniel Dirkschmidt in Freiburg at Roche Pharmaceutical Company, and you also went to Dr. Bergmeier in Chapel Hill in the USA. And I also found back your motivation letter from April 2019. And there you wrote, "My passion is understanding inflammation and cardiovascular research, and I'm eager to invest my strengths and skills into a new challenge. I have proven my abilities to think critically and prioritize work and communicate requests and research subjects in my prior work. I aim to contribute to my future team's work, to grow professionally and personally, and I'm highly motivated to start my scientific career in the field of cardiovascular research and inflammation." So obviously, to us, your team, you were actually the best candidate for our position, and we really moved quickly before you would be like hijacked by other members of the consortium to choose another project. So during the past 5 years, you have more than delivered on your promises. Although all can see that you are small in stature, especially compared to me, you have proven that you are actually a person of big value and great personality. I have gotten to know you as a committed and persistent researcher, but also as a very nice and social person who fits well into our group and department. You organize several enjoyable group activities, such as a visit to the Museum of Illusions, which is very nice. And you learned your way around the platelet lab pretty fast, as well as familiarize experiments involving cell culture. On the other hand, you did not like to be dependent on other people for getting things done. This could be either experiments or data analysis, but also administrative procedures, which can sometimes take a while at the University of Maastricht. And that tested your patience in the first few months, as I remember. But I really admire your talent for prioritizing work and setting your own boundaries for what you can and cannot do. On several times, you clearly communicated to your supervisors when you would be able to work on what and when you would be not available. And this was something I had not seen in a PhD student before. Unfortunately, the COVID pandemic came about 5 months after you started. And during that time, all trainings and meetings from the consortium were online, as well as all the lab meetings. And this was actually a quite difficult time for you, as you were new in Maastricht and did not like working alone in your room without any personal interactions. So as soon as work was again allowed in the offices, you came back in as one of the first, because even if it was only for a few hours. So during your PhD period, you moved and traveled a lot. You presented your work at several international meetings and also performed experiments in other labs. In September 2021, you went to ESAS in Dortmund to perform all the proteomics measurements. And these were quite complex and difficult, but you persevered with the help of Professor Siegmann and especially Dr. Fiorella Solari. And directly after ESAS in 2022, you moved to Mainz in Germany, close to your hometown, for a secondment of 1 year, where you were supervised by Dr. Jörg, present here, and also Professor Walter. And I really recall some nice visit of me and Professor Heemskerk to Mainz to either perform experiments or discuss the project and enjoy some nice dinners. And also we met online almost every month with the Maastricht, Mainz and ESAS team to discuss all the progress of your projects and also of Pengyu Zhang, the other PhD T-cardio student that we supervised. So in January 2023, you moved back to Maastricht for the remainder of your PhD project. It was a great pleasure for me to be your supervisor. You were always motivated and came up with new ideas for experiments and how to continue with projects. Of course, there were also some stressful times, especially during the COVID time, but you hung in there and kept going, which finally resulted in this excellent thesis and the outstanding defense you did today. So you have really proven to us that you yourself are also a true jack of all trades. You can be very proud of yourself, of what you have achieved, because for sure I am very proud of you.
Thank you.
I would like to end with thanking the members of the assessment committee, Professor Ten Kate, Professor Dankwart, Professor De Meijer, Dr. Nuels, Professor Ori, and Professor Reinhardt for their time to evaluate and approve the thesis. I also would like to thank all of those present here, as well as Professor Schaefer and Dr. Roest for coming here to Maastricht and participating in the Corona. This ceremony could not have taken place without you. Dear Claudia, I wish you lots of success in your upcoming career and all the best for the future. I'm very happy that you will stay in our research group as a postdoc, and I look forward to keep working together. Enjoy all the beautiful moments that are yet to come, and start today because it's your PhD celebration. So I give the word back to the pro-rector.
Well, we come to an end. So first I would like to say, the congratulations, Dr. Schaefer, how does that feel?
Good.
Yes. So Dr. Schönichen, I would like to say congratulations also on behalf of the board of Maastricht University. In my congratulations, I also would like to include the promotion team who guided you since 2009, 2019, I would say that's the better frame. I also would like to thank basically the members of the Corona. They were already mentioned that they could join you today. I mentioned sometimes you have 7 minutes to ask questions, but it takes more than 7 minutes to prepare these questions. And I would like to thank you for that time. I would like to also say congratulations to your family. Und wir machen das in gutes Deutsch, dass wir sagen, schöne Grüße und schöne Glückwünsche zu der Familie hier. We say also thank you very much here who are attending this thesis defense. It was a good thesis defense, and I can tell you that we came to a unanimous positive decision and there was no negative sign, so you made a good impression. And that is time now also then for some celebrations. So we're going to celebrate this now in the rafter, but before we start that, I'm going to use my hammer, which you haven't seen, which I'll show you right over here. And with this hammer, I officially close this ceremony. And how do we proceed? We're going to make a picture here of the members. University, and then we will also join you in the so-called rafter. That's the place where you can have some coffee, some beer and some wine. And you can already start over there, and we will join you in about 7 to 8 minutes from now. Thank you.